5 minutes. This is the first time I've run through this one. I decided today, instead of going into my thesis work, to tell you a story about conservation. It's a really cool story. It's something that I just became a part of. We just became a part of this really neat conservation story that's sort of globally important, and it has to do with frogs, and it has to do with human health, and it has to do with evolution. So it goes all the way around the world, starts in Africa, comes all the way back, and instead of going through methods and materials of sort of what my thesis is about, I thought this story would be a lot more interesting. But first, we'll start with how I got involved in Drexel University and conservation to begin with. Back in the early 90s, my advisor, Dr. Berner, traveled to a little island off the coast of West Africa called Bioko. Bioko has a really unique set of primates. Gail was going there with a group of other primatologists to search for, among others, the elusive drill monkey. They found the drill along with a host of other really endangered, unique primates, and thus started the Bioko Biodiversity Protection Program. This has been Gail's project for the last 20 years, and when she moved to Drexel, she took on graduate students, and we started working and studying biodiversity conservation on the island. It's a really unique island. It's located just off the coast of Cameroon, about 35 miles. It's got 3 major peaks on it. It was a continental island, meaning it was connected to the mainland, so it has the full host of biodiversity that you would expect on the mainland, as opposed to an oceanic island that would have to be colonized over time. So because of this, it was separated, it was protected, while the rest of the land on the mainland was sort of destroyed over time. So it's a really important place for conservation, and also a really easy place for evolution if you're studying island biogeography. And to the point of the mountains, you have a very small island with a lot of relief. So you have a lot of different habitats in a very small area. So you have a lot of different microhabitats. So you have the opportunity for really, really high biodiversity in a very small area. This is just an example of the different habitat types you're going to see on Bioko, from the coast to where the top of my studies range around 2,000 meters above sea level. It's a biodiversity hotspot, so I don't know if you can see this map right here, but biodiversity hotspots around the world. If you look there on West Africa, the West African forest, in the Gulf of Guinea, we're right in there. The Bioko Island is right in the middle of that biodiversity hotspot. It's also part of the Cameroon volcanic line. It's a volcanic line that ranges up through Cameroon and Nigeria. And what's neat about the Cameroon volcanic line is that these high elevation habitats are like islands in and of themselves. Just like you think of Madagascar being very unique and endemic, you have the high elevations of these islands are also unique, evolve unique and endemic species. So Bioko is part of that. We see a lot of endemism on those islands. You also have the separation from the mainland, so divergent evolution on Bioko. A lot of opportunity for really unique biodiversity. These are the sorts of things that we see when we walk around on Bioko. We spend about 2 to 3 months a year there, our lab. I spend a lot of time with primatologists, so they're walking around looking up. I'm walking around looking down. We run into each other often. We also have some sea turtles. 5 species of sea turtles nest on the beaches. One of the most unique places in all of West Africa for sea turtle nesting. So all in all, the take-home message for Bioko is that one of the most important places for biodiversity conservation in all of Africa has a really unique set. Most of these photos were from National Geographic, who came to the island during one of our expeditions a few years back. But we're concerned with amphibians, right? So why study amphibians? Amphibians are really, really cool, first of all, but they're also really important. Ecologically important. They're sort of what you might consider a keystone species. And again, to avoid sort of ecological terms for this broad crowd, you think of it as a keystone species, everything is reliant upon it. So everything eats frogs. Frogs eat a lot of insects. Tadpoles eat a lot of algae. So everything is connected to frogs. You pull them out of the ecosystem and things start to fall apart. They're also indicators of ecosystem health. The first things to die when an ecosystem is polluted, or when we see climate change, the first things that start to get affected are amphibians. They're a model organism for studying evolution, and in the past 10 to 20 years, we've seen an increased rate of extinctions, mass die-offs in frogs, and that's sort of the story we're going to be talking about in a minute. So for a lot of reasons, amphibians are really ecologically, evolutionarily interesting organism to study. Again, global amphibian species richness around the world. If you can see that, the red areas are the highest diversity all over the world. So sort of the Amazon, obviously you have incredibly high species richness of amphibians. If you can see where Africa is right there, right where Bioko Island is in the Gulf of Guinea, also one of the highest areas of amphibian richness. So if I didn't convince you before, Bioko is really, really important. Not just for biodiversity, but specifically for amphibian ecology, evolution, and conservation. So, Bioko amphibian research, again, I've gone through most of these unique montane habitats, high diversity. The one unique thing about studying frogs in Bioko is that there's nothing there to eat you. I talk and I work with a lot of other herpetologists who go out and they sample it. You can only catch frogs at night. They sample at night, but they only sample for a half an hour, 10 minutes because there's forest cobras or elephants or leopards or lions or whatever other thing you have on the mainland. Luckily, Bioko doesn't have any of those things, so we can go out all night and we can catch a lot of amphibians in a small amount of time. And to that point, in the 4 years I've been there, we've discovered up to 4 new species, and those are them listed, pictured right there. 2 large-bodied tree frogs, this small leaf litter frog, and a divergent species that on the mainland it looks a lot different. I think it's going to be. So, again, Bioko's a pretty neat place. High rainfall, lots of steep rivers and canyons, so a lot of places for them to be isolated within the island and diverge. We talked about this story of mass amphibian die-offs. How many people have heard of chytrid? So not even half of you. So, which is understandable. Chytrid is not a very well-known disease. It's been around for 20 to 30 years now. It swept right through North America. It's sweeping through South America, and it's a fungal disease that kills amphibians. It's wiped out up to 30% of all the 6,000 known amphibians are at risk of amphibians. It's up to 400 species are either experience mass die-offs or are at risk of extinction currently. It's one of the largest vertebrate die-offs we've ever seen in modern history. It's a really, really big deal, and nobody knows about it. And like I talked about before, you pull amphibians out of an ecosystem, and things start to collapse. It also, we don't really know where chytrid came from. It's a complicated story. There's multiple strains of it. We just sort of started detecting where these strains are coming from, but we're really not sure. And one of the most, the hardest hit areas for chytrid are these high montane areas. So again, Bioko Island is going to be really susceptible. High diversity, tropical area, and montane habitat. This is a recent map. This was just published. It's just published in January. This is sort of a synthesis of all the different records of chytrid. So now what people are doing, they're going out, they're doing presence assays of chytrid, and they're finding where chytrid is. And if you can't see this map, look for the red dots. The red dots are the presence of chytrid. White dots and blue dots are where people looked and they couldn't find it. You look at Africa, and there's not much. There's a few dots in East Africa, a lot of blue and white, so we don't really either know much about chytrid in West Africa, or we haven't been sampling there very often. This map, this figure, I'm not going to dwell on too much, but again, you see a lot of dots in North and South America. That's where the research is focused. That's where a lot of people who do research, ecological research live. So that's one reason. But again, Africa, not very much work going on for chytrid research. What's unique is that Africa could be the origin of Chytridiomycosis. back in the 1930s and 40s, the first pregnancy test that we ever developed was from something called Xenopus laevis. It's the African clawed frog. What they did was they injected it with a human woman's urine. The woman was pregnant. The frog started to ovulate. If she wasn't pregnant, it didn't. It was our first pregnancy test. And so the African clawed frog was mass exported all over the world from West Africa. And they think, one of the theories is that chytrid was on the backs of these African clawed frogs. And that's how Xenopus spread all over the world. So, why is there an apparent absence of chytrid in Africa? Is it just a sampling bias? Does Bioko Island have chytrid? Despite positive surveys in some parts of Africa, why are no mass die-offs reported? And if we find chytrid, will it support this out-of-Africa hypothesis? I was able to attend a lecture here at Drexel by Dr. Reed Harris. And this was, up until this point, we really had no way to stop chytrid. There was the only thing that people were doing is pulling frogs out before chytrid got there, keeping them in zoos away from the areas that were affected so that they could maybe later on reintroduce them. Dr. Harris was studying salamanders and found that the bacteria on their skin produce metabolites that are antifungal in nature. So certain species they found have resistance to chytrid. He found this salamander in North America had resistance. He worked with Dr. Kevin Mimbiel over at Villanova now, and they characterized the bacteria and the metabolites from that bacteria, and they found that that's what offered resistance. So now we finally have a clue. We have a little bit of hope. Here's the mechanism. Basically, bacteria on the skin of salamanders produce these antifungal metabolites. The chytrid persists, but it can't overtake the organism, so it persists on the skin, but it doesn't survive. So they developed a probiotic, basically a bath of bacteria that could be beneficial for these frogs. They just ran this experiment. They put a bunch of frogs that were susceptible to chytrid. They were dying of chytrid. They put them in these little containers. They shook them around in the broth. They let them go, and sure enough, it worked. So this is hope. Finally, after 10 years of amphibian extinctions, we see one technique that might actually hold some hope in the future. So we have a really interesting part of the story in Bioko. We're in West Central Africa, where Xenopus is from. We might have frogs that are resistant to chytrid because they co-evolved there. So our study sought to find out if chytrid's in West Central Africa, and characterize those bacteria and metabolites. Because we haven't seen mass die-offs, we have high diversity. So if chytrid's there, they're probably resistant. If we can figure out what's on their skin, we can actually provide a really important puzzle piece to the probiotic solution for chytrid. This is an example of one of the habitats that we sample in. Samples were taken in the wet and the dry season. We studied over 400, we sampled over 400 individuals, far more than most of the other studies in Africa. So there was no sampling bias where we were. Sure enough, Bioko Island has chytrid, quite a bit. Each one of those pie graph results indicates how many samples. So if it's bigger, there's more samples from that location. Red is positive, blue is negative. So Bioko has a lot of chytrid, but despite the fact that over 95% of the individuals and species were positive for chytrid, they all had non-lethal infection loads. They had genome equivalents. 12 was the highest, and it was 12 or less, average about 4. You need an infection load, you need a genome equivalent of 10,000 to be lethal. So, again, support for out of Africa, support for these guys being resistant. So, and this all happened just in the last year. So we decided, well, let's keep going. We went back to Africa and we swabbed again. This time we swabbed for metabolites and bacteria. And this chart we actually made 2 days ago. We just got this data back. And it doesn't mean much to you until I do that. These are, by color, different species. And that is the group of metabolites per species. So what this means is each species has a unique grouping of metabolites on their skin, which also could mean they have a unique community of bacteria on their skin. That's really neat. That's evolutionarily significant. That has conservation implications. And then this paper came out. And this paper basically says, yes, it's really important. Peptides metabolites play a very important part in it. When we saw this paper, that's what we did in the lab. That means that our work is really important. It's going to play a really important part of the puzzle. So implications, Yoko has Chytrid, we've confirmed that. Low infection, low possible resistance supports the Africa hypothesis. The metabolite analysis and the initial bacterial analysis say these things are distinct to the species. That's really neat for coevolution. It's really neat for trying to figure out the probiotic. In the future, we're going to try and characterize the bacteria and the metabolites. I know I've gone over time. Mitch, I apologize. So I'd like to thank quite a few people, my committee, the Bern Lab, all my collaborators, and funding for this project, hopefully into the future, because we have some interesting stuff. And before I leave you, I'm going to do one more plug. Our lab recently put together a documentary, and it's screening here at Drexel for the first time on April 15th from 6 to 8. Feel free to come by and learn about primate conservation on the Oval Island.
It's not easy being green. Again, waiting until later for questions. Now we have our third and final presenter, Derya Meral, who, I take the laundry out of my dryer and I fold it, right? But she folds proteins, which to me is like, and not only that, virtual proteins. So no proteins were harmed in this work. So just without further ado, Derya.